everyone, a very welcome to Vyas Edification Kota. So today in the series of NCERT discussion, we are going to discuss a new chapter from class 12, that is molecular basis of inheritance. This chapter contains total 14 questions and each and every question will be going to discuss in this session. The very first question of this chapter is on the screen. Group the followings as nitrogenous bases and nucleosides. We have given adenine, cytidine, thymine, guanosine, uracil and cytosine. Among these followings, we have to identify which of these are nitrogenous bases and which are nucleosides. So, before going on to this, we have to know that what is DNA. We know that DNA is a polymer of nucleotide. It is a polymer of nucleotide. And what are nucleotides? These are the units which is made up of sugar, nitrogenous bases and a phosphate group. So all these three things, phosphate group, sugar and nitrogenous bases, constitute together to form a nucleotide unit of a polymer of DNA. Now, we know that nitrogenous bases are included in the nucleotide of a DNA. So what are these nitrogenous bases? Basically, in the DNA, we have 4 bases. What are these 4 bases? Adenine, guanine, thymine and cytosine. So clearly, we can identify this adenine, this thymine, this uracil and cytosine. All these are the nitrogenous bases, whereas we have left with 2 things, that is guanosine and cytidine. These are nucleosides. But what are nucleosides? We know that a nucleotide is a mixture of 3 things: phosphate group, sugar and nitrogenous bases. If a nucleotide is present without phosphate group, we will call it as nucleoside, because it is not having phosphate group. So what are nucleosides? Guanosine and cytidine. So let's write answer of this question. If we talk about a DNA, then a DNA is a polymer. DNA is polymer of nucleotide. DNA is a polymer of nucleotide which are made up of sugar, nitrogenous bases, and a phosphate group. Basically, we have 4 bases. Basically, DNA have 4 bases. What are these? These are adenine, thymine, uracil and cytosine. So all these are the nitrogenous bases. Whereas what is nucleoside? A nucleotide without phosphate group is known as nucleoside. Now, which of these are nucleoside? Obviously the cytidine and guanosine. These are the nucleosides. Guanosine and cytidine. So This is the differentiation between nitrogenous bases and your nucleosides. Nucleoside is whenever a nucleotide lacks phosphate group, we will call it as nucleoside. Right, so this is the complete solution of question number 1. Now let's talk about next question. If a double-stranded DNA has 20% of cytosine, if in a double-stranded DNA there will be 20% of cytosine, then we have to calculate the percentage of adenine in the DNA. We have given that in a polymer of DNA, which is a double-stranded DNA, the percentage of cytosine, which is a nitrogenous base, is given as 20%, and we have to calculate the percentage of adenine in the DNA. So how we will calculate this? We have Chargaff's rule for this. So according to Chargaff's rule, all the cellular DNA regardless of species, number of adenosine residues is equal to the number of thymidine residues. That means A is equal to T. And the number of guanosine residues is equal to the number of cytidine residues. That means G is equal to C. And we have given C as 20%. So automatically if C is 20%, then G will also be 20%. And to calculate the E, we will subtract the total percentage of C and G from 100%. So let's write the answer of question number 2. First of all, we'll write this like according to Chargaff's rule, according to Chargaff's rule, all the cellular DNA, regardless of species, number of adenine residues is equal to the number of thymidine residues. That means A is equal to T, and the number of guanosine residue is equal to the number of cytosine is equal to the number of cytosine residues. So we have given now according to this, here also G is equal to C. According to Chargaff's rule, in any cellular DNA, regardless of species, always A will be equal to T and G will always be equal to C. Now we have given cytosine as 20%. So obviously the G will also be 20% because G is equal to C, right? And C is equal to G. Both are equal to each other. So we have given 20%, automatically G will also be 20%. Now to calculate the percentage of adenine, we know that we have to subtract this. So to calculate adenine, 100 minus G plus C. We will do 100 minus G plus C, and we know that we will minus 40% from 100. We will minus 40% from 100%. We'll get 60%. But this is not the answer because this 60% is the number of residues of both adenine and thymidine. Now what we have to do to calculate percentage of adenine, this 60% is going to divide by 2 and we'll get 30%. So 30% is adenine and the 30% is thymidine. Alright, so this is the complete solution of question number 2. So let's discuss next question, question number 3. If the sequence of one strand of DNA is written as follows, that is 5 prime, and we have given this sequence ATGC, 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 ATGC and ATGC and 3 prime. Write down the sequence of complementary strand in 5 prime to 3 prime direction. So according to complementary base pairing, we know that always A pairs with T and C pairs with G and vice versa. So we will write the complementary sequence of this given strand, that is 3 prime. We are writing the complementary sequence of this given sequence. So this will start with 3 prime and we know that A pairs with T, T pairs with A, G pairs with C and C pairs with G. Similarly, we'll write T, A, C, G, then again T, A, C, G, then again T, A, C, G, T, A, C, G, then again T, A, C, G, again T, A, C, G, again T, A, C, G, then again T, A, C, G and 5 prime. So this is the complementary sequence of the given strand. Now we have to write this complementary strand, this complementary sequence of a strand in 5 prime to 3 prime direction. So we will write from here to here in the 5 prime to 3 prime direction. 5 prime and starting from G, C, A, T, G, C, A, T, again 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 G, C, A, T, and the last G, C, A, T, and this is 3 prime direction. So this is the sequence of a complementary strand. This is the complementary strand or the sequence of a complementary strand with 5 prime to 3 prime direction. And this is a complete solution of question number 3. Now let's discuss question number 4. If the sequence of coding strand in a transcription unit is written as follows, that is 5 prime ATGC, 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 ATGC and 3 prime. Write down the sequence of mRNA. So we have given the base sequence of a coding strand of DNA. Now what is coding strand and what is template strand? We know that in DNA is a double-stranded molecule. DNA is a double-stranded molecule. One strand of DNA which is participating in the synthesis of mRNA is termed as template strand, whereas the other one strand which is not participating in the formation of mRNA is termed as coding strand. Now the base sequence of coding strand of DNA is similar to the base sequence of newly synthesized mRNA. Always the base sequences of coding strand of DNA and mRNA are same. Hence we can call it as the mRNA is identical to the coding strand except for the uracil in place of thymine. And we will write the answer of question number 4, and at the end of this answer we will write the sequence of mRNA by using the sequence of this coding strand. This is given as coding strand. Now answer will start like this. A DNA is double stranded molecule. The strand of DNA which is participating in the synthesis of mRNA is template strand, the other strand of DNA, is coding strand, which have the similar base sequence with mRNA, Alright, the base sequence of coding strand of DNA is similar to the base sequence of mRNA. So mRNA is identical to the coding strand except for uracil, in place of thymine, in mRNA. So if we want to write the mRNA sequence by using this coding strand, we will write like this. We will write it as 5-A. We will write U in place of T. So AUGC, then again AUGC, then again we have AUGC, again we have AUGC, again we have AUGC and AUGC, and 3'. Always the sequence of mRNA will be identical to that of the coding strand of DNA, and the thymine will be replaced by another kind of base, that is uracil, in case of mRNA. So this is a complete solution of question number 4. Now let's talk about the next question, that is 5th question. Which property of DNA double helix led Watson and Crick to hypothesize a semi-conservative mode of DNA replication? Explain. So to answer this question, we know that Watson and Crick were the two scientists who observed that the two strands of DNA are present in anti-parallel direction and have opposite polarity. They were the two scientists who observed the anti-parallel arrangement of two strands of DNA. Also, they observed that the two strands of DNA are present in the anti-parallel arrangement and have opposite polarity. That means the 5' phosphate of one strand of DNA always faces the third hydroxyl group of other strand of DNA. Also, 5' phosphates of both the strands are present in opposite direction or opposite position. Now, this anti-parallel arrangement of two strands of DNA allows hydrogen bonding between amine and carbonyl group of the complementary base pairs. This led them to hypothesize the semi-conservative mode of replication, wherein the two strands of DNA first separates from each other, followed by copying of the two strands, both the strands of DNA, to form two molecules of DNA. Each molecule of DNA will contain one parental strand and one newly synthesized strand. So this is the answer, and we have to write the solution, which will start like this, that Watson and Crick Watson and Crick were the two scientists who observed that the two strands of DNA are anti-parallel to each other and have opposite polarity That means the 5' phosphate of one strand faces the third hydroxyl group of other strand and the phosphates and the 5' phosphates of both strands are present in opposite position. This thing led these scientists to hypothesize the semi-conservative nature of or mode of DNA replication. Wherein what happens in DNA replication and why we call the DNA as semi-conservative in nature, because in the DNA replication the two strands of DNA first separates from each other, followed by copying of both template strands of DNA, to form two molecules of DNA. Each molecule of DNA will have one parental strand and one newly synthesized strand. This is why we call the DNA as semi-conservative in nature, because during the DNA replication, the two strands of DNA separates from each other, then they copy both the template strands to form two molecules of DNA. Each DNA molecule then will consist of one parental strand and the one newly synthesized strand. This thing led the scientists such as Watson and Crick to hypothesize the semi-conservative nature or mode of DNA replication. So this is the complete solution of question number 5. Now we have question number 6 on the screen. Depending upon the chemical nature of the template, DNA or RNA, and the nature of nucleic acid synthesized from it, DNA or RNA, list the types of nucleic acid polymerases. Now on the basis of chemical nature of template strands, we basically have two types of nucleic acid polymerases, that is DNA dependent DNA polymerase, and the another one we have is DNA dependent RNA polymerase. So let's write answer number 6. We have two types. Basically we have two types of nucleic acid polymerases. What are these? First, if we talk about, that is DNA dependent DNA polymerase. These are the enzymes. So if we talk about the first one, that is DNA dependent DNA polymerase, this polymerase enzyme adds the nucleotides to form or to synthesize DNA by using the base sequence of parental strand or template strand of DNA during replication. So this is one type of nucleic acid polymerase. Then the next type of nucleic acid polymerase we have is DNA dependent RNA polymerase. Right, now this enzyme or this polymerase enzyme adds the ribonucleotide primer strand to form RNA by using the template strand of DNA during the process of formation of RNA. So this is a complete solution where we have two types of nucleic acid polymerases. One is DNA dependent DNA polymerase, and the another one we have is DNA dependent RNA polymerase. DNA dependent DNA polymerase adds deoxyribonucleotides to form or to synthesize DNA by using the parental or template strand of DNA during the process of replication, whereas the another type of nucleic acid polymerase, that is DNA dependent RNA polymerase, this is also an enzyme, polymerase enzyme, which adds ribonucleotides to form or synthesize RNA by using template strand of DNA during the process of RNA synthesis. Now this is the complete solution of question number 6. Now moving on to the next question, that is question number 7. How did Hershey and Chase differentiate between DNA and protein in their experiment while proving that DNA is the genetic material. Alright, these were the two scientists, Hershey and Chase were the two scientists who cultured bacteriophages in two different medias. These both scientists cultured bacteriophages in two different medias. One media is containing radioactive phosphorus and the another one media of bacteriophages containing radioactive sulfur. Now, these scientists allowed these bacteriophages to infect their host cells, that is Escherichia coli. Now we know that phosphorus is the content of nucleic acid, is the content of DNA which forms the nucleic acid, and the sulfur is the content of amino acids such as methionine and cysteine, which are the amino acids among 20 amino acids which forms protein. Now after infection, they take the samples for analyzation and they separated these bacteria and viruses with the help of centrifugation. Now we know that bacteria are larger in size, so they formed a solid pellet at the bottom after centrifugation, and phosphorus or the viruses remained in the supernatant of the centrifugation. Now after analyzation, the host cells or bacteria are found to be radioactive when they infected with radioactive phosphorus, whereas no radioactivity was found in the host cells or Escherichia coli or bacteria which was infected with the radioactive sulfur. This proves that the DNA is actually the genetic material. Now let's write the answer of this question number 7. And it will start like this, that Hershey and Chase, these were the two scientists who cultured the bacteriophages in two different medias, in two different medias. One is containing radioactive phosphorus, and the other one is containing, the other media is containing the radioactive sulfur, Now we know that phosphorus is the content of DNA, and sulfur is the content or constituent of amino acids such as methionine and cysteine, which are the amino acids among 20 amino acids which forms protein. Now the scientists such as Hershey and Chase allowed these samples, or we should not write samples, they allowed these they allowed these bacteriophages, which are viruses, bacteriophages to infect the host cell, that is E. coli. That is E. coli, which is a bacteria. So they allowed these bacteriophages, which are the virus infecting bacteria, to infect the bacteria such as E. coli or Escherichia coli. After infection, for analyzation, they separated the bacteriophage or viruses, bacteriophage and E. coli, in which the E. coli forms a solid pellet at the bottom, and viruses, because they are smaller in size, viruses remained in the supernatant, right? Now, after observation, what they found, that the bacteria or the host cell, bacteria or host cells are radioactive when infected with the radioactive phosphorus, and no radioactivity was there in the bacteria or host cells when infected with the radioactive sulfur, which proves that DNA is the genetic material. And on the screen we have a written solution for this question. Who find difficulties in writing the answer, long answers in English? This written solution is for those students. In this solution also, the same thing is written, that Alfred Hershey and Martha Chase, which is the complete name for these two scientists, they cultured bacteriophages in two different medias. One is containing radioactive phosphorus and another media is containing radioactive sulfur. Now, we know that phosphorus is the component or content of nucleotides, which are DNA, which forms a nucleic acid such as DNA, while the sulfur is the component or content of amino acids such as methionine and cysteine, which are the amino acids among 20 amino acids which helps in the formation of protein. Now, these two samples of bacteriophages were allowed to infect their host cells such as Escherichia coli or E. coli, which is a bacteria. Now, after infection, the samples were analyzed for the radioactivity in the host cells, of course. For analyzation, what they did, they separated the bacteria and viruses via centrifugation. Since the bacteria are large in size, they form solid pellet at the bottom of after centrifugation, and smaller viruses remained in the supernatant after configuration. The bacterial pellet was found to be radioactive when infected with the radioactive phosphorus or the bacteriophages with radioactive phosphorus, and there was no radioactivity found in the bacteria or Escherichia coli which were infected with the bacteriophages with radioactive sulfur. Now, this things proves that the DNA is actually genetic material. DNA is the genetic material, not proteins are the genetic material. And on the screen you can see a diagram. This is the experiment 1, this is experiment 2. In the experiment 1, we are testing the proteins. We are testing proteins with the help of radioactive sulfur. Now see, this is a bacteriophage, a virus which is able to infect any bacteria. So this is a bacteriophage. The scientists radioabled the protein codes of this virus and allowed this bacteriophage to infect the bacteria such as E. coli or Escherichia coli. Then they analyzed it and no radioactivity was found in the Escherichia coli when it was infected with the bacteriophage which was labeled with radioactive sulfur. But in the experiment 2, when these scientists radioabled the DNA of this virus or bacteriophage and allowed this bacteriophage to infect the Escherichia coli or bacteria, after analyzation they found this Escherichia coli or E. coli was radioactive. Alright, so from this experiment they proved that genetic material is DNA but not proteins. This is a complete solution of question number 7. Now let's discuss next question, that is question number 8. We have to differentiate between repetitive DNA, satellite DNA, and then we have to differentiate between mRNA and tRNA. Then last we have to differentiate between template strand and coding strand. So let's start with the very first thing, that is differentiating between repetitive and satellite DNA. What is repetitive DNA? The non-coding sequence on DNA, the non-coding sequence which is present on DNA and repeated several times in the genome of an individual is termed as repetitive DNA. Whereas the satellite DNA is one of the various categories of repeated DNA which contains highly repetitive sequences. Now depending upon the number of repeats, satellite DNAs are grouped into various categories such as mini satellites, microsatellites and mini variant satellites. So this is the difference between repetitive DNA and satellite DNA. Repetitive DNA is nothing but non-coding sequences present on DNA which are repeated several times in the genome of an individual. And satellite DNA is one among them which is containing highly repeated units or highly repeated bases. Then come towards the differences between mRNA and tRNA. So mRNA is the messenger RNA. mRNA specifies amino acid on the polypeptide. mRNA specifies amino acid on polypeptides. Thus, it is transferring genetic material from DNA to ribosome, whereas tRNA, which is also known as transfer RNA, reads the genetic information which is present in the triplet on mRNA. tRNA reads the genetic information which is present in the triplet, which is present in the form of triplets on mRNA, and transfers the appropriate amino acid from the amino acid pool of cytoplasm to the growing polypeptide chain during protein synthesis. So this is the difference between mRNA and tRNA. The last one is we have to differentiate between coding strand and template strand. We know that DNA is a double-stranded molecule. The strand of DNA which is participating in the process of formation of RNA, that means in the process of transcription, that strand of DNA which is participating in the transcription process is known as template strand, whereas the another one which is similar in the base sequence of mRNA and is not coding for any RNA is termed as coding strand. So let's write the answer of 8. First we will differentiate between repetitive DNA and satellite DNA. So the non-coding sequence of DNA which is repeated several times in the genome of an individual, in the genome of an individual is repetitive DNA, whereas the satellite DNA is one of the repetitive DNA which contains highly repeats. Now, depending upon the number of repeats, depending upon the number of repeats, satellite DNA is grouped. Satellite DNA is grouped into various categories such as mini satellite, microsatellite, and mini variant satellite. Now, if we write the differences between mRNA and tRNA, so we will write the difference between mRNA. mRNA is the messenger RNA, which specify the amino acids on a polypeptide, That means, thus, it transfers the genetic material from DNA to ribosome, whereas tRNA, that means transfer RNA, reads the genetic information on the genetic information which is present in the form of triplets on mRNA, right? So this is the difference between mRNA and tRNA. Then last is the difference between template strand and coding strand. So we'll write the difference between template strand and coding strand. Template strand is the strand of DNA which participates in the transcription process that is formation of mRNA from DNA, whereas the coding strand is the strand which do not codes for anything, and is similar to mRNA except for uracil. uracil. Alright, so these are the differences between template strand and coding strand, and the differences between mRNA and tRNA, and the difference between repetitive and satellite DNA. With this, we have completed the solution of question number 8. Now let's discuss the question number 9. List 2 essential roles of ribosomes during translation. We have to give 2 essential roles of ribosomes during the process of formation of RNA, that means translation. So the 2 roles of ribosomes are as follows. First of all, ribosomes act as the site where the protein synthesis is taking place, from single or individual amino acid. So first function or first essential important function of ribosome is it provides the site for protein synthesis. Now these ribosomes are made up of 2 subunits, the smaller subunit and the larger subunit. The smaller subunit of ribosome binds with mRNA and forms protein synthesizing complex, and the larger subunit of ribosome acts as the amino acid binding site. Now, the second function of ribosome is it acts as catalyst during the process of formation of polypeptide bonds. So these are the 2 essential roles of ribosomes, and we are going to write the answer number 9. The two essential roles of ribosomes are as follows. The first function is the ribosomes acts as site where the protein synthesis is taking place. From an individual amino acid. So first essential function or role of ribosome is to provide the site for protein synthesis. Also, if we talk about the ribosome, then the ribosomes are made up of 2 subunits, the larger subunit and the smaller subunit. The smaller subunit of ribosome binds with mRNA and forms protein synthesizing complex. And the larger subunit acts as amino acid binding site. This is the first role of ribosome. Then the second essential role of ribosome is it acts as catalyst. It acts as catalyst during formation of polypeptide. So these are the 2 essential roles, 2 essential functions of ribosome, and with this we have completed the solution of question number 9. Let's come towards the next question, that is question number 10. In the medium where E. coli was growing, lactose was added, which induced the lac operon. Then why does lac operon shut down sometimes after addition of lactose in the medium? So we know that lactoperon is active in the presence of lactose. Lactoperon is active in the presence of lactose, and whenever the lactose is absent, the repressor binds to operator and do not let the genes to be transcribed. Now, this lactose when present, it binds to the repressor and it codes for some enzymes such as beta glycosidase, permease and trans-acetylase. The enzyme beta glycosidase converts the lactose into glucose and galactose, which was readily available and readily used by the cells. Now, whenever the lactose is present or lactose is added, the lactose once gets used, the lactose is completely used, the lactoperon shuts down, because in the absence of lactose, the repressor binds to operator and do not let the genes to be transcribed. The permease enzyme, which was encoded by the lactoperon, enhances the permeability of cell membrane so that lactose is transferred into the cell once it is available. So, low level expression of lactoperon is always required. Now, let's write the answer. Answer number 10. We know that lactoperon is active in the presence of lactose, in the absence of lactose, the lactoperon do not let the genes to be transcribed. But whenever the lactose is present, it will bind to the repressor. So, in the presence of lactose, it binds to the repressor. And encodes the enzymes such as beta glycosidase, permease and trans-acetylase. So, whenever the lactoperon is added, after complete usage of lactose, the repressor shuts down and do not code for anything, because repressor in the absence of lactose will bind to the operator and this does not lead to the formation of these enzymes. Now, this beta glycosidase, what is the function of beta glycosidase? It converts the lactose. Whenever the lactose is present, the lactose will bind to repressor and forms these enzymes. After the formation of these enzymes such as beta glycosidase and permease and trans-acetylase, these beta glycosidase converts the lactose into glucose and galactose. glucose and galactose, which is readily used by the cell, and the permease enzyme enhances the permeability of cell membrane. This is the function of enzyme permease. So it enhances the permeability of cell membrane so that the lactose is transferred, into the cell once it is available. Once the lactose is available, it can easily transfer into the cell because of the increased permeability of cell membrane with the help of this permease enzyme. So always low level expression of lac operon is always required. Why it is always required that lac operon should expresses and forms these enzymes? Because after formation of these enzymes, permease enzyme will enhance the permeability of cell membrane so that once the lactose is available, it can easily transfer into the cell. So this is the complete solution of question number 10. Now let's talk about the next question, that is question number 11. Explain in 1 or 2 lines the functions of the following. And we have to explain the functions of each of them, such as promoter, tRNA and exons. So what are the functions of promoter? Promoter is a site present physically continuous with transcription unit and serves as the binding site for RNA polymerase enzyme. So this promoter is actually providing the binding site for the RNA polymerase enzyme. This is the function of promoter. If we talk about the function of tRNA is also known as transfer RNA. What is the function of tRNA? It reads the genetic information. Which is present in the form of triplets on mRNA, and also it transfers the appropriate amino acid from the amino acid pool of cytoplasm to the growing polypeptide chain. This is the function of tRNA. If we talk about the function of exons, then these exons are the sites, these exons are the protein sequencing sites which actually transfers the genetic information from DNA to protein. So exons are the sites which is providing genetic information from DNA to protein. So let's write the answer 11A. We're writing the function of promoter. So the promoter is the site present physically with transcription unit, and serves as the site to RNA polymerase enzyme. So what is the function of promoter? It provides the site for RNA polymerase enzyme. Next, if we talk about the function of tRNA, which is also known as transfer RNA, then the function of this RNA is to read the genetic information. It reads the genetic information present in the form of triplets on mRNA, It reads the genetic information in the form of triplets which are available on the mRNA, and also transfers the appropriate amino acid, from amino acid pool of cytoplasm to the growing polypeptide chain. So what is the function of tRNA? It transfers the amino acid from the amino acid pool which is present in the cytoplasm of a cell. It takes the amino acid from there and it transfers that particular appropriate amino acid to the growing polypeptide chain. So this is the function of tRNA. Let's talk about the last one, the function of exons. Functions of exons. So these are the protein sequences which carry the genetic information from DNA to proteins, So basically it carries the genetic information. Exons are the sites which carries the genetic information from DNA to protein. So these are the functions, and with this we have completed the solution of question number 11. Now let's talk about question number 12. Why is the Human Genome Project called a mega project? Why is the Human Genome Project is known as mega project? So Human Genome Project was a mega project because there was a sequencing of each and every nucleotide base pair present in the human genome, which took around 13 years of time for its completion. The aim of this project was to develop new technologies and new information in the field of genomic studies or in the genetics. This project was a large-scale project. The Human Genome Project was a large-scale project which provided the new information, which provided the new opportunities in the field of genetics, biotechnology and medical sciences. Also, this project provided some clues for understanding the human genome. That is why we call this Human Genome Project as a mega project. So let's write answer of this, answer number 12. It will start from this only. Human Genome Project. Human Genome Project is a mega project, because there was the sequencing of each and every nucleotide base pair present in the human genome. Base pair present in the human genome. Alright, so this was a mega project because there was sequencing of each and every nucleotide base pair present in the human genome. The aim of this project, was to develop new technologies and information new information and new technologies in the field of genomic studies. This project, this human genome project, was a mega project, was a large scale project. So this project was a large scale project, which provided the information or new opportunities in genetics, biotechnology and medical sciences and it took around 13 years for its completion. This was so huge that it took around 13 years for its completion. Also, this project of human genome provided some clues to understand the human genome. So with this, we have completed the solution of question number 12. So let's talk about question number 13. What is DNA fingerprinting? Also mention its application. So what is DNA fingerprinting? DNA fingerprinting is simply a technique which is used to identify or analyze the variation on the basis of variation and polymorphism present in DNA sequences. This is DNA fingerprinting, and if we talk about the applications of DNA fingerprinting, which are as follows. The DNA fingerprinting is used to identify the crime suspect. The DNA fingerprinting also can be used in paternity testing, and also it can be used in to finding out the evolutionary history of an organism. So this is answer number 13. What is DNA fingerprinting? DNA fingerprinting is simply a technique. It is simply a technique in the medical sciences, which is used to identify or analyze the variations, on the basis of variations and polymorphism in DNA sequences. The applications of DNA fingerprinting are as follows. The first application of DNA fingerprinting is it is used to identify the crime suspect suspect. Also it can be used, also it can be used in paternity testing, in paternity testing. Then it also can be used to find out the evolutionary history of an organism, evolutionary history of an organism. So this is the complete solution of question number 13, in which we have covered the definition or the meaning of DNA fingerprinting, which is simply a technique which is used to identify or analyze the variation depending upon the variation in polymorphism present in the DNA sequences. And we also have seen the applications of DNA fingerprinting. It is used to identify crime suspect. It also can be used in paternity testing, and it also can be used in, to find out the evolutionary history of an organism. With this, we have completed the solution of question number 13. Now moving on to the last question of this chapter, that is question number 14. Briefly describe the following terms such as transcription, polymorphism, translation and bioinformatics. So we'll be discussing each and every term, or we'll be explaining briefly, describing briefly each and every term on the screen one by one. First we are taking the transcription. So we have to describe transcription. First of all, what is transcription? Transcription is a process by which RNA is synthesized from DNA. So simply the transcription is a process in which RNA is synthesized from DNA. Transcription is a three-step process. What are these three steps? Steps are included in the process of transcription, that is initiation, elongation and termination. In the process or in the step of initiation, what happens? The RNA polymerase enzyme binds to the specific sequence of DNA which is known as promoter and provides the single stranded template strand of the DNA during the process of transcription. Now in the process or in the step of elongation, this RNA polymerase enzyme reads the template strand of DNA and adds bases to elongate the chain. In the last step of transcription, such as termination, specific terminating sequence marks the end of transcription process. So this is how RNA is going to be made from DNA, and the process of formation of RNA from DNA is termed as transcription. So let's write answer number 14, and we'll be writing part A of this, that is transcription. Transcription. So what is transcription? It is a process of formation of RNA from DNA. Simply it is a process of formation of RNA from DNA, and we call it as transcription. It is a three-step process. It is a three-step process. What are these three processes included in the transcription? That is initiation, elongation and termination. So first step of this transcription process is initiation. In this step, what happens? RNA polymerase enzyme binds to the promoter and providing binds to the promoter, which is providing the single stranded template during transcription. The next step of this process, transcription, is elongation. Now in this step, RNA polymerase enzyme RNA polymerase enzyme reads the template strand and adds the basis to elongate the chain. Then the third last step of this process transcription we have is termination, in which a specific terminating sequence marks the end of this process, marks the end of this transcription process. So this is the description of part 1 or part A of this question number 14. We have described the transcription process, which is a 3-step process. We have covered all 3 steps of this process. This is the complete solution of part A of question number 14. Now let's discuss the B part of this question, that is we have to describe about polymorphism. What is polymorphism? Polymorphism is a kind of genetic variation. Which leads to the formation of several different kinds of individuals among the members of one particular species. What is polymorphism? Polymorphism is a kind of genetic variation. It is nothing, it is a form of genetic variation which results into the formation of several different kind of individuals among the members of one particular species. Now, the varying sequences of nucleotides are located on the different positions of DNA helix. The varying sequences of nucleotides are located on the different positions in a DNA helix, which result into the variation and polymorphism in a population. This polymorphism is responsible for speciation and evolution as well. So let's write the B part of this question. What is polymorphism? Polymorphism is a form or kind of genetic variation which leads to the formation of several different types of individuals, among the members of one particular species. Now this varying sequence of nucleotide are located on different positions in a DNA helix, which results into variation and polymorphism in population. Population. This polymorphism also responsible. This polymorphism is also responsible for speciation and evolution So this is the complete description of polymorphism, and with this we have completed the B part of question number 14, and moving on to the next part, that is C part, in which we have to describe about translation. So now let's discuss translation. What is translation? It is also a process. Now, translation is also a process in which the protein is going to be synthesized from RNA. Translation is basically a process through which protein is synthesized from RNA. Now, this translation is also a three-step process. What are these three steps? Initiation, elongation and termination. Just similar to the transcription process, here also in translation we have three steps such as initiation, elongation and termination. In the process of initiation in translation, what happens? The ribosome assembles, or the ribosome attaches to mRNA, which has to be read, and the first tRNA will be carrying the amino acid such as methionine. Then in the next step, such as elongation in translation, what happens? The amino acid chain is being elongated. Then in the last step of translation, that is termination, the synthesized polypeptide chain is being released. So these all are the three steps included in the process of translation, which we are going to write on the screen now. Translation. It is a process of formation of protein from RNA. So simply, translation is a process from which we will synthesize protein from RNA. It is also a three-step process. The very first step of this translation is initiation, in which the ribosomes are attached to the mRNA and has to be read. And the first tRNA in this process will be carrying the amino acid methionine. This is the first amino acid which will be carried by the first tRNA in the process of initiation of translation. Then the next step under this process we have is elongation. Now what is elongation? In the step, the chain of amino acids is being elongated by the addition of several amino acids. It is being elongated. The chain of amino acids is being elongated by the addition of several kinds of amino acids. The last step we have is termination. In this process, the synthesized polypeptide chain is being released. So in the process or in the last step of translation, that is termination, the newly synthesized polypeptide chain is being released. So this is the complete description of a process translation. With this, we have completed the third part or C part of this question. Now we'll be discussing the last part of this last question, that is description of bioinformatics. So what is bioinformatics? It is the use of information technologies. Bioinformatics is the use of information technologies for collecting and analyzing biological complex data such as genetic codes and protein sequences. There has been an advancement in the field of molecular biology. There is the invention of computers, scientific techniques and instrumentations at the level of nano level. So these have helped in the development and progress of bioinformatics. So let's write the last part of this question. Bioinformatics. So bioinformatics is the use of information technologies. Bioinformatics is the use of information technologies for collecting and analyzing the complex biological data, and etc. with the help of databases. Now there has been an advancement. In the field of molecular biology. There is invention of computers, scientific techniques and instrumentations at nano level. These have helped, all these have helped in the progress and development of bioinformatics. So this is the complete solution of D part of this question, and with this we have completed all the questions included in this chapter. This is all for today. Thank you and have a nice day.